Hi, it's Miss Barnhart. Let's go over some basic genetic terms and how to do Punnett squares. First, let's take a look at some helpful vocabulary. You really need to make sure you know these terms in order for you to do well in this unit. First, a trait is an inherited characteristic. A gene is a segment of DNA that codes for that trait. An allele is different forms of a gene. A dominant allele will always express itself when present. A recessive allele can be masked by that dominant allele. The genotype is the allele pair combination. Homozygous is if the two alleles are identical. Heterozygous if the allele pairs are different. Phenotype is the physical expression. So let's take a look. If capital A is the dominant allele, lowercase a would be the recessive allele. Big A is homozygous dominant. Big A little a would be heterozygous dominant, or we would just say heterozygous. Little a is homozygous recessive. And those are my genotypes. But big A and big A little a both show the dominant phenotype. Little a shows the recessive phenotype. Now let's take a look at Punnett squares and how to solve genetics problems. There's my Punnett square. And in my monohybrid cross, here's what I do. I want to set up a key, and I want to clearly identify the dominant and recessive allele. Secondly, I want to identify the parental genotypes from the information that's given to me and write that down. Next, I'll figure out the gametes that can be made, and I want to write those down the sides and top of the Punnett square. I'll combine those gametes, and I'll fill in the Punnett square would be the zygotes, and then I determine my genotype and phenotype. So take a look. If I'm looking at axial stems, which are dominant to terminal stems, I'm using a big A and a little a. My problem is crossing two heterozygous plants. That would be big A little a times big A little a. Here's my Punnett square. Each one of the parents can make a big A and a little a in their gametes, so both the egg and the sperm, and I write down the side. Then when I fill in big A, big A, big A, little a, big A, little a, and little a, that's what's going to be in my zygote. Notice that I have 1, 2, 3 of the genotypes, the big A and the big A little a, can both show, all three will show the dominant phenotype. So I have a 1-to-1 genotype ratio and a 3-to-1 dominant to recessive phenotype ratio. Now, what happens if I cross a heterozygous plant with one with terminal flowers? So that's big A little a times little a little a. There's my Punnett square. This time, notice that one plant can make big A little a in the gametes. The second one can only make little a alleles. So big A little a, big A, little a little a, big A little a, little a as I fill in these squares. Here we're going to have a 1-to-1 dominant to recessive genotype and phenotype, where there would be one half of the plant would have axial stems and the other half would have terminal stems, full terminal flowers. So a test cross is when I can check to see what my unknown phenotype is. Notice the white flower is always recessive, but my purple flower could either be homozygous or heterozygous dominant. So how would I know? The way I do this is with a test cross. I want to cross my unknown genotype with a homozygous recessive plant. So in this case, I would cross the purple plant with one that's homozygous white. If any of the offspring end up purple, if any of the offspring end up white, then the plant's heterozygous. If all the offspring are purple, then it's most likely